Biowaivers for modified release dosage forms non-B strengths. This is the very important topic for the present video, and I welcome all the viewers. I request to pay your 100% attention to each and every slide of this video so that you can understand this topic in a great way. Modified release formulations may be sustained release, prolonged release, delayed release, or these may be having the biphasic release. The modified release formulations contain some excipients which can modify or which can tailor the release of the active from the formulation. And mainly this video is for the oral formulations which have modified release. These formulations may be tablets, capsules, these may be the pellets as modified release formulations. Modified release formulations have different advantages like reduced dosing and reducing the pill burden. Some therapeutic advantages can be taken from the modified release formulations. MR formulations or SR formulations contain or sometimes may contain higher dose compared to the immediate release formulation. For modified release formulations, biowaivers are not granted, but the non-B strains can get the biowaiver. Always understand that for modified release formulations, direct biowaivers are not possible because these are the formulations which are critical for the therapeutic benefit and the pharmacokinetics are altered by modifying the drug release for such formulations. Let's see about the product specific guidances provided by different authorities. I have enlisted the example from reference of the product specific guidelines by some regulatory authorities. So this PSG provide you the bioequivalence requirements and also the information on biowaivers. Generally, I observed that for modified release formulations, B study is performed on strength which may be higher strength, middle strength or lower strength depending on to the formulation and the molecule class or the drug therapeutic class. And on the B strength, the bioequivalence study is performed in fasting condition and also in fed condition. The design may be crossover design or it may be a parallel design based on the half-life of the molecule. Then for some of the formulations, multiple unit dosing is also done or steady state study can also be done. And waivers are given for the lower strains or the strains which are non-B strains. Also, in some of the guidelines, you can observe that complete B study in fasting and fed is performed onto the highest strength and only fasting or only fed bio study is required or B study is required in only fasting or fed on the lower strains. That means bracketing is required. For example, if the fasting is a critical condition to have bioequivalence, then higher strength will be studied for both fasting and fed, and lower strength will be studied in fasting only, so that the regulatory authority can get the correct information on the bioequivalence. Along with these bioequivalence studies, the regulatory body or regulatory authorities require discriminatory resolution. Also, comparative dissolution profile is to be done in different medias with the pH range of 1.2, 4.5, 6.8, and in some cases along with the QC release media. So profiling is required. Then alcohol dose dumping study is required to meet for the modified release formulations. And also sometimes other studies may be required as per the guidelines of the regulatory authorities. Sometimes the modified release formulations may be present or designed in a MUX type of formulation, that is multi-unit particulate system, in which the multi-units of pellets are compressed into a tablet formulation. So for these formulations, break line can also be provided onto the tablet, and for these type of formulations, split tablet dissolution is required to be performed. If the formulation is dose proportional or dose weight, then you can ask for the biowaiver for the lower strengths. But it depends onto the molecule therapeutic class. Also, it depends onto the formulation design, type of polymer used, number of strengths, and some other critical criteria. Now, see the biowaiver considerations for SR formulations or modified release formulations, and the main focus is on the rate controlling polymers. Which type of polymers are used, how much quantity is used in the formulation to control the drug release. What is the design of the formulation, whether the design is same for all the strengths or it is somewhat different among the strengths. Also, we need to understand the qualitative and quantitative composition of the MR formulation or SR formulation with different strengths. Proportionality of rate controlling polymer across strengths. So generally we know that, for example, hydrophilic polymer containing tablets or matrix tablets contain higher polymer in the lower strengths if the API is soluble, like metformin, and the proportionality is difficult to meet. So lower strength qualify for biowaivers if polymer to the drug ratio remains unchanged. Biowaiver possible if dissolution profiles remain similar. That means F2 criteria should apply and the lower strengths should be with passing F2 criteria to the higher strength dissolution. Regulatory agencies like USFDA, EMA require evidence that polymer changes do not impact the release kinetics. Generally, it is observed in hydrophilic type of tablets. The lower strengths may release the API at a faster rate compared to the higher strengths. So, the release controlling polymer is present in higher amount in the lower strengths compared to the higher strength formulation. SUPAC guideline considerations are also there, which are need to be critically evaluated. There are three changes: level 1, level 2 and level 3, based on to the changes, whether the change is minor, major or moderate or major. Level 1 change in minor polymer adjustments within or below 5%, so no impact can be expected. Level 2 is between 5 to 10%. It requires additional dissolution studies. Significant polymer changes come under the class or level 3, where polymer level is more than 10% change, and this requires the bioequivalence studies to support the biowaiver for the lower strengths, and also IVIVC is required. Now, impact of polymer on the drug release mechanism. If polymer type and grade are consistent across strengths, biowaiver is feasible. If polymer content is reduced, validation through IVIVC or multi-point dissolution profiling is required. Generally, the polymer quantity may change, but the polymer viscosity and other critical material attributes of the polymer should be same. Examples of polymer influence or impact like hydrophilic polymers, HPMC is there. It maintains the gel matrix integrity. Hydrophobic polymers like ethyl cellulose, it controls the diffusion rate. The modified release formulations or sustained release formulations may be monolithic or may be the coated formulations. These may be the pellets or these may be the muffs type of formulations. SUPEC considerations include viscosity changes and the polymer substitutions. So we need to understand the chemistry of the polymer, quality and quantity of the polymer among the strengths. Dissolution testing and IVIVC for justification. Multi-pH dissolution studies are performed to confirm the consistent drug release. Generally, dissolution is done with the media pH 1.2, 4.5 and 6.8. Some other medias may be required based on to the drug properties. Discriminatory dissolution studies are required to identify the formulation differences. IVIVC models confirm reduced polymer levels do not affect the drug absorption kinetics. Strong IVIVC may justify a biowaiver without in vivo studies. So in this, PBPK modeling can also be done. Dissolution data is required to be presented. There should be a discriminatory dissolution report whenever you are applying for the modified release type of formulations and asking for the waivers. Then regulatory challenges and considerations. Generally, direct scaling down of polymer proportion is preferred, but many of the time it doesn't work for some of the molecules which are highly soluble, like metformin hydrochloride. If polymer content is significantly lower, a biostudy may be required, and that's why sometimes the product specific guidelines require one biostudy on the lower strength and complete biostudy on the higher strength. Risk assessment crucial for BCS class 2 and 4 drugs with solubility concerns. If the formulation contains low soluble API, then the biowaivers for lower strengths also become very critical. Regulatory precedent strengths the case if RLD biowaiver is approved. So there may be a strengthening of the case for biowaiver in the regulatory environment or in the regulatory authorities. If you provide the data or reference that the RLD has got biowaiver approval for the lower strengths. This happens if the RLD is applied through 505(b)(2) pathway, and if the innovator or NDA applicant has got biowaiver for the lower strengths. Literature support and post-approval changes guidance can help you for the justification. Conclusion for the biowaiver for lower strength for SR formulations. These require proportional polymer reduction without affecting the release kinetics. Similar dissolution profiles across the strengths. IVIVC validation supporting formulation changes. C. If polymer changes exceed SUPAC limits, bioequivalence study may be required. Regulatory harmonization enhances access to cost-effective SR formulations. So, in summary, understand the BCS class, understand the design of the formulation and polymer level. Also, understand the release kinetics, and then based on these things, you can think for applying for the biowaiver for lower strengths. For higher strength or B strength, bio study or bioequivalence study is required to be performed. Whenever you are developing the defined release formulations or sustained release formulations, you should do the multimedia dissolution study, CDP studies, that is comparative dissolution profiling studies from initial phase of the product development, so that there will not be any surprises at the time of exhibit batches and filing. Also, make the process robust and do the study for CMS of the polymer, CPPs of the process, and effect of the stability. This is regarding the biowaivers for modified release formulations. I hope you will get good idea for this topic from the video. Thank you.